So, hello everyone. We are once again in what we are reading section of the South American Mycorrhizal Research Network for our YouTube channel. Today is a very exciting day because I don't know if you know about this recent article which I will share with you at one moment. The article is by Justine Karst, Melanie Jones, and Jason Hoeksema. Is called Positive Citation Bias and Over-interpreted Results Lead to Misinformation on Common Mycorrhizal Networks in Forests. This article is, in my view, a critical article in mycorrhizal ecology. Hopefully it will change a lot of things, a lot of research agendas. And we are lucky enough today to have the three authors of the article. So if you can please introduce yourselves. Justin.
Hi, everyone. I'm Justine Karst. I'm an associate professor at University of Alberta and the current vice president of the International Mycorrhizal Society.
Melanie.
I'm Melanie Jones. I'm a professor of biology at the University of British Columbia's Okanagan campus. And I've been working on mycorrhizae for about 40 years and mycorrhizal networks for about 25, 30 years.
Wow. And Jason.
Hi, I'm Jason Hoeksema. I'm a professor of biology at the University of Mississippi.
Great. So as mentioned before, this article goes against many outlandish claims about common mycorrhizal networks, examines these claims critically. So my first question to any of you will be, what was the motivation behind the paper? What was the motivation behind this huge literature analysis that you did?
Yeah, I can start. For me, as you mentioned, Cesar, the extraordinary claims that I was hearing, initially I thought, I didn't remember the studies. I didn't remember coming across the studies that supported some of those claims, but I assumed that I just wasn't on top of the literature. And so the extraordinary claims, and then also for me, it was when my son, who was in grade 8 at the time, he came home and he told me that he learned that trees were talking to each other through below ground fungal networks. He learned this at school. And to me, that was the instance I also realized how widespread these claims were. And it was then that I had started talking with Melanie and Jason, and we decided to tackle this review.
And I just wanted to add from my perspective, when Justine approached me and was asking about the literature in the CMN field, was I've been aware for quite a long time that some of the key papers that had been published were not being cited especially accurately, in that some of the results that weren't significantly different were being reported as if they were, and also that many of the alternative explanations, not all of them, but I was aware of a few of them at the time, that they weren't being included when people were citing the papers.
Guille, go ahead, please.
Yeah, well, I'm very excited to be here. I really like the paper. I really admire these researchers. So one thing that I came across while preparing the questions is that the paper presents and offers a lot of definitions, which I think is great, because you need to first present the concept and then see what is the limitations. So I may ask for some couple of definitions that I think that are key to understand the paper. The first one can be, if you can briefly explain the Wood Wide Web concept or theory, and then if this, because I find some doubts or some claims that this paper will finish this theory, and I don't think so. But if you can try to explain it.
Melanie, do you want to take that one? That comes from your paper.
Yeah, I guess I'm not even sure what you mean by the theory, because where the Wood Wide Web term came from was from the original Simard et al. Paper 1997 that was published in Nature on the front cover. And I was one of the co-authors. I was involved in that study. And when Nature decided to put that on the cover, the editors at Nature came up with that term, Wood Wide Web. So that's the only context in which I've thought of it, is just that it was the title that was put on the cover of Nature. So I'm not even really sure what you mean by the theory of the Wood Wide Web. It's just a term that I see out there. And that was part of our intention, was to show people what evidence are there in terms of mycorrhizal, common mycorrhizal networks in soil.
Mm-hmm. And then what I understand, I just recall a tweet from a Spanish guy asking in Spanish, saying that this might be the end of it. But then what I understand of the paper is not this, is that you are trying to know what we know about this and how much we can generalize, right? So that is one of the main contribution, opening the door to the things that we don't know. And we have to know, considering some of the limitations that you have analyzed. Is that correct? I don't know.
I mean, Jason might want to talk about some of the definitions, because I think you're right that it's some of the terminology, some of the terms are used in different ways by different people, and I think that is adding to some of the confusion.
Yeah,
Absolutely.
and I guess I would just say that the Wood Wide Web, I think, is somewhat equated to the idea that there are mycorrhizal connections between trees and forests. Common mycorrhizal networks, CMNs is the acronym often used. And I want to be clear that what we were examining was the evidence for the structure and function of those CMNs. We're not commenting on the science or the value of mycorrhizal fungi for trees. One definition we try to clarify in the paper is that mycorrhizal networks in general, that term is often used to just refer to mycorrhizal fungi extending as a network into the soil from the roots of a single tree, whether or not it's connected to another tree. We were examining common mycorrhizal networks that are shared between different trees, and the evidence for how widespread they are and how persistent they are, and then especially for their function. Do they benefit the trees? Do they transfer resources enough that it has consequences for the fitness of receiving trees? We were also not commenting on CMNs between trees and herbaceous plants. For example, mycoheterotrophic plants, for which there is good evidence that mycorrhizal networks, common mycorrhizal networks, transfer important amounts of resources from trees to these mycoheterotrophic plants. The claims that are out there associated with the Wood Wide Web are really about tree interactions through these common mycorrhizal networks. So that's what we focused on in the review.
You focus on three main claims, and I don't know if you have them in mind, and you can briefly explain the claims, and we can go briefly through all of them.
Sure, I can start. The first claim, so first of all, I should back up. So those claims, where they came from, is we reviewed a lot of popular media, books, newspaper articles, documentaries, TED Talks, all the stuff that the public interfaces with, to extract what we thought, these are the claims we see over and over and over again. So that's where those three claims came from. And the first claim is so the first claim is that common mycorrhizal networks are widespread in forests. And as most of all your listeners will know, we can't dig in the soil to see the common mycorrhizal network, because when we do it breaks the network, it disrupts the network. And so we thought the strongest evidence, the best evidence, was the maps that have been done, the genotype mapping. And there's only 5 such maps done, and 4 of them are interior Douglas fir forests, and then 1 of them is in a pine forest in Japan. And 2 of them we think are very convincing in showing that it's very likely that roots are connected by the same genet. But that's only 2 studies of the 5. And then, of course, then there's only 5 studies done in 2 forest types. So to make this claim that common mycorrhizal networks are widespread in forests, we don't think that we know yet. There's a lot of uncertainty. But I think everybody would agree it's very likely that trees are connected below ground through these fungal links, but we don't know how long those connections last. We definitely don't have a good idea about their function. And so that was our conclusion for claim 1. I can turn it over to Melanie to talk about claim 2.
Sure. So the second claim was that there's resources that are transferred between trees within the hyphae, and it benefits seedlings. And so we spend quite a chunk of time in the paper talking about the different experimental approaches that have been used to try to study whether or not there's, say, carbon or nitrogen or water moving between trees using these networks. And that's where I really will refer people to the papers. The key things is that as we discussed the papers, and the 3 of us met weekly and really reviewed the papers in general together in depth. And it was so interesting because each of us had some ideas already about some of the confounding factors, but there were more of them that we all came to realize as we read the papers. And we thought, oh my heavens, that paper that I published however, a few years back, there's this other possible explanation for it. So I'll leave the listeners to go and read that in some detail. I think that the part of the claim that I always like to focus on is the part about, do these physical connections benefit seedlings? And when we looked at the experiments that had been done in forests, because we didn't really think that the lab studies were relevant for looking at that particular claim, seedling survival or seedling growth, did either of those improve in treatments where there was the potential for a network to form? The other important thing is that it was almost never that experimenters, including ourselves, tried to determine whether or not a mycorrhizal network actually did form in the treatments that we called the CMN treatments. When we looked at all of the studies that we could find, all of the experiments, we found that in 18% of them, we saw that seedling performance improved in the treatments where the potential for the CMN was there, and that benefit was retained in situations where roots were also allowed to interact. So only 20%, or less than 20%. In the other 80, the seedlings either showed no response to the potential mycorrhizal network, or they actually showed a negative response. And so, one of the really strong messages is, and I know this is a point Jason often makes, is we need to understand why there seems to be an improved seedling performance in some cases in response to CMNs, and in others it's negative. In other cases, it's neutral. The results were so variable, and we couldn't attribute them to any particular environmental factor that was measured.
I have a couple of questions. First, for Justine, you mentioned that 5 articles, 5 studies use genotyping, right, for see how widespread or not are these common mycorrhizal networks. So did they sequence like a specific gene, or it was whole genome sequencing?
Yeah, no, good question. I believe in all 5 of those studies, they were using microsatellites. So in those systems, they were single species stands. So in Douglas fir, pretty much all the trees were Douglas fir. And Melanie, please correct me if I have any of this wrong. Melanie's husband, Dan Durrell, was involved in a number of these mapping studies. So they could dig in the soil and then find roots. And the roots that were colonized by rhizopogon make very distinct ectomycorrhizal structures, like little potatoes. So you can see, oh, okay, that's rhizopogon. And rhizopogon only forms these structures on Douglas fir roots. And so when you collect that, you can then genotype the roots and know what stem they belong to, what individual tree. You can genotype the fungus because you know it's a rhizopogon, and then using the microsatellites, you can tell which genet it belongs to. So is this genet different from this genet colonizing another root system? So I'm pretty sure those studies were, I mean, it's been a little while, but yeah, I'm pretty sure they all use microsatellites to get that information.
So what would be a better method or a better sequencing technique if we want to do like these global maps? Should we use
Yeah.
backby or something like that, or what should we do?
Oh, that's a good question. I haven't quite thought that far in like what is the next sequencing step? I mean, sequencing has changed and developed and improved a lot in the past 10, 15 years of when some of these studies were done. And I think whatever sequencing method we choose, it needs to be able to identify genotypes. So not just species of fungi or roots, but you also need to be able to go down to the level of genotype. And this is important because only genotypes will fuse. If they're different genotypes, different species, they won't be able to form those common mycorrhizal networks. Yeah.
And I think that some of the other omics approaches will be useful, like some of the metatranscriptomic or transcriptomic approaches, because that will give some insight into which parts of this network are functioning. And I think that's the part that we're really missing in terms of these maps.
And for Melanie, you mentioned that just in 18% of the studies there was a benefit to seedlines, but a big part of your article was that this benefit can be caused, the explanation can be other than the common mycorrhizal network. So why is that? Why the explanation can be other than the network?
Well, those are two separate points, I would say. One is just to be very clear that regardless of the mechanism of interaction, there's only 18% of the seedlings that show any increased survival or growth. The alternative explanations so there's really sort of two groups of studies. There's ones that look at how did seedlings grow in response to these different treatments, and then there's a group of other studies that just look at isotope movement of isotopes of carbon or water or nitrogen, and there's very few that look at both. So the critique about let's think about other possible explanations for mycorrhizal function was referring to the second group, the group that was of studies that was looking at the movement of the isotopes. And I guess there were maybe 3 or 4 confounding factors, but that's where I know you want us to be brief. So I would refer people to the paper. It has to do with root interactions, has to do with the volume of soil that is being manipulated and constrained in some of the treatments, and also that the communities of fungi, both mycorrhizal fungi and pathogenic fungi, could be altered by the treatments, and we hardly ever look at that.
Cesar, can you put the figure 1? You are mute.
Yeah. We have 10 more minutes, so yeah, go ahead.
So I was thinking while we were talking about this, here is the first figure of the paper. I really like it, and these are the columns to the right are the different positions of the different alternative explanations, and there are many that deals with pathogens, for example, or fungal, or even access to surrounding roots. So I have just one quick question about that some of the papers didn't consider, and I think it was Justine mentioning that sometimes the root can be there, or the so the transfer of nutrients from mature trees to seedlings can happen because the mature roots are intermingled with the seedlings. So there is no need, or maybe there is no need of common mycorrhizal networks. Is that an alternative hypothesis that what is called, is this what is called this soil pathway? Like the nutrients can be transferred through soil, so there is no need for common mycorrhizal networks.
Well, really, there are, I would say, three main pathways that, and I think one of the questions that we would argue should be focused on going forward is just understanding how it really works when resources are transferred between trees, if they are, and what are the relative contributions of different pathways. So one possible pathway is a continuous common mycorrhizal network pathway. One of them is that roots may transport resources long distances and be right up against the roots of another tree. And then there's a bulk soil pathway where through water in the soil, resources may move. And the relative combinations of those different pathways are what we really should try to quantify. In many of these studies, the distinction between that soil pathway and the mycorrhizal network pathway was not clear, or there was often evidence that in fact the soil pathway may have been quite significant relative to the common mycorrhizal network pathway, and that was not often acknowledged strongly enough. Or the original authors did acknowledge that, but then that gets ignored in subsequent citing of those papers or reference to those papers in the popular media, because that's a distinction that is kind of swept under the rug. But we would argue that those kinds of distinctions are important for really just understanding how the whole system works. And then it's a whole nother question that, what are the consequences of that resource flow for the performance of plants? And that's often not a piece that is also added on.
Yeah, in the same figure, all the studies that you're presenting in rows might present alternative hypotheses, meaning that there is no, and I was very surprised to see that there is no single study that has unequivocal evidence of this functionality of common mycorrhizal network.
It's difficult to pinpoint the role of the mycorrhizal network, in resource transfer, seedling performance. But as you mentioned, in some of those papers that are in that table, in the discussion, they do mention the caveats, the limitations, alternative explanations. But as Jason said, they don't always get carried forward. And that is why we did the citation analysis, where we see evidence for bias. But before we get there, I want to leave time for Jason to talk about claim 3. Yeah.
Right. Well, claim 3 is the idea that trees signal when they're being attacked by insects or other pests, and they signal to their offspring or other genetic relatives about that attack, which allows increased performance or survival and upregulation of defenses in those recipient trees. And for this claim, we found very, very little support, especially in field studies. We found that, in fact, when you look at the support that's being referenced in these popular claims, it's pointing back to just a couple of greenhouse studies, which have fascinating results, but not results that point clearly to this particular claim, in fact. So just to give one example, there's a greenhouse study by Song et al. From 2015 that is a very nice study. The results from that suggest that when Douglas fir is attacked by insects or clipped to simulate attack, there is evidence for upregulation of defense enzymes in nearby pine seedlings when mycorrhizal networks are connecting those seedlings, but only when roots are not allowed to naturally intermingle between those adjacent seedlings. In a different treatment where there's the potential for networks to form between fungi, but also for the roots of the adjacent seedlings to intermingle, that effect goes away. There's no evidence for signaling and upregulation of defenses. And it's that latter treatment with roots intermingling that really mimics reality and what's really happening in a forest. And so to use that to extrapolate all the way out to real forests and say that this is what trees are doing, adult trees are doing among each other in a real forest, is a stretch that we can't make if we're really trying to base our claims on science.
That brings me to a question that I had, which is how to combine these two approaches, because I totally agree that fieldwork studies are vital, but sometimes to disentangle different factors, you need laboratory. And I think that this opens this huge discussion between these 2 points of view. So how to match those two things? What do you think?
I guess I would just say that I think in order to study, say, seedling performance, you need to do that study in the field. Anything where you're looking at something that a large tree is doing, of course, you have to do that in the field. And then what I would argue is if you see a response, and then you want to understand the mechanism of the response, that's when you can try to design a lab experiment to test that. But we shouldn't take results from the lab that say show growth responses or something like that and apply them to the field. It should be the other way around.
Guille, one last question that maybe you have. We have a couple of minutes.
Yeah. There are many. It's very rich paper. There is a lot of information about this alternative hypothesis. I invite the listeners to read carefully. Yeah, I wonder, as the numbers and the percentage of the functionality, for example, of the common mycorrhizal network are surprisingly low for someone that I was not working on these networks, but I really believe, and this I think is a proper verb here. Do you think that I tend to think that maybe as we haven't been able to demonstrate, besides all the limitations of all these studies, that the common mycorrhizal networks are functionally very positive in a way, do you think, and I'm referring here to a blog from Matthias Willig saying that maybe we have focused too much on fungi and maybe they are not so important and we're missing some other organisms. And I was wondering what you think about that.
Well, I mean, all of us are interested in fungi because, we study mycorrhizal ecology. So we will come to it with that view. But no, I think it's a good point. There's lots going down. Like there's lots of organisms that are doing things at that scale. And to focus on one particular element, I mean, we could scale up and say, even in the forest, do we just focus on this one part? It's probably not going to tell us the whole story. But we get trained in subdisciplines and we have to focus. But no, I take his point. I think it's bang on. Yeah.
One last question that I have for you is about the functionality of the possible common mycorrhizal networks. We established that so far there is no evidence that they are improving seedline performance, right? So what we consider, what would be the time frame considered so the common mycorrhizal network is a functional network? So for how long it should be established? Because I also imagine that many like accidental connections are established, but they are also quickly breaking down. So for how long it should be this connection?
Yeah. So for me, that reminds me. So there was only one study, and it was done by Marty Kranebetter, where he set up a mesh experiment in a typical way, trying to manipulate access or not to mycorrhizal networks. And it was the one study that I think was done for 5 years. And in that study, depending on what time point you looked at, the potential effect of common mycorrhizal networks changed over time. After 2 years, it looked kind of positive, but then later on, it definitely looked negative in that they were not helping the seedlings. So I guess that's sort of on a different scale, but most of our experiments, they're done over 1 or 2 years. And of course we have to do that because the seedlings grow big, you know, they're only in these little tiny bags. And so we don't want to be measuring the response to the size of the bag. So there's that issue, I think, and just that we're looking at very small snapshots of time. And then I think, Cesar, what you're asking is just also about more on the micro scale of like these connections might form, they might break. How long does it have to be a connection for it to count, to be functional for the seedlings? And those are really good questions. Maybe Melanie and Jason have an answer, but I'm not sure. I'm not, yeah.
Well, I guess what I was going to say was, you know, again, I think we're falling into a trap of thinking about things from the plant perspective. And so a CMN, I think it's very easy to comprehend how beneficial it would be for a fungal genet to be associated with a number of different trees and receiving carbon from different trees that might be, you know, in slightly different physiological states or whatever. So I just want to put a little plug in that we shouldn't forget that aspect as well.
Yeah, I have a last question, and this is related to maybe part of the title, or maybe one of the main messages of the paper is about the positive bias. The paper presents a very thorough review of the evidence of the common mycorrhizal networks, and to me is very convincing and very strong. And this makes me think that how can, if we can use this, or we can, this paper can help us to think how much evidence we need to tell, for example, the journalists how safe or how are our messages, how uncertain or certain are our messages. I don't know if you have thought about that, but do you have any thought on this?
Jason, Melanie, I have some thoughts. So I think it's not, so with the citation bias, yes, there were instances where researchers would, you know, speculate too far or generalize too much. But it seemed to be more of an issue of extracting particular claims or results as evidence for pushing this story that common mycorrhizal networks are beneficial to trees. They move resources around. It seemed to be more in that direction. And so one thing, while we were doing interviews, one of the journalists asked us, because they were also surprised that when it all came down to, say, claim number 3, there's a couple of greenhouse studies. That's it. And that's quite surprising. And they asked, you know, so when you're talking to journalists, what can journalists do so this kind of thing doesn't happen again? And then Melanie had this really nice suggestion of journalists asking, so yes, your results are really interesting, they're highly novel, but are there alternative explanations? And it's a great question to ask scientists and to ask ourselves in terms of, you know, we want to see stories sometime. You know, we all have hypotheses that maybe we're wedded just a little too tightly to, and it blinds us to alternatives. And on the media side, if they ask those sorts of questions, it forces the scientists and the journalists to think, what else might be going on? How certain? What's the next step? What else do we need to know? And I think that simple question for scientists and journalists would really take us far in terms of representing the scientists, representing the uncertainty, and tamping down on the hype.
Yeah, thanks a lot. I think that was the last question, right, Cesar?
Yes, to your last point, Justin, that's something that I really like it about the paper, that invites to look for alternative explanations, as any good scientist should do. And also, it's not a confrontational paper. I see more confrontation in the news media, but the paper is quite straightforward in its arguments. So I really like that, and thank you to all of you, to the 3 of you, and to Guille, for this space. And if you have some last thing you want to say, please do.
Thanks for the opportunity to talk about it. Appreciate it.
Yeah, thank you very much for giving room for our voices. Thank you.
Thanks a lot.
Yeah,
Thanks
thank
to all of
you.
you.
Nice to meet you. Bye.